This is now your pre-lab. We are doing your pre-lab right now. Notice, did you get a thick packet?
No.
All right. No thick packets, now it's on you. Grab paper, calculator, grab paper, colored pencils. Be ready to do some work when you come in tomorrow. You'll need your goggles tomorrow, yes. Gloves are going to be required as well. All right, it says a setup procedure, it's there. Biochemical test, it's called paper chromatography. It's on that page, it's in a column, left hand side, it says paper chromatography. Does everybody see that? Now, one of the things that we do in science, one of the things, let me tell you a story. Kids like stories, so. There's these ancient group of people known as alchemists. Do you know what these things were? These people were? They were the founders of the thoughts of chemists, yeah. But alchemists had one purpose in mind: to turn lead into gold. They wanted to take lead, which you could find everywhere, and just turn it into gold. They felt that through a combination of magic and science, they could make lead turn into gold. On the way to their search for this enriching process, they discovered all kinds of things. And they started the foundation, what we call chemistry today. Obviously, it wasn't chemistry. It was kind of a hybrid process. Alchemists were all over the world. One of the things they always found, one of the things that they developed, one of the things that we've been doing as humans forever, is separating things. So you have this tube of stuff. Somebody has you spit in a test tube. They take your blood. It's a solution, some kind of solution. The thing that everybody wants to do is separate it. How do we get what we want out of this and leave the rest? The miners of the old 49ers, you ever hear Miners 49ers, that old song? The gold rush, you've heard of the gold rush? Yeah. People would go in the streams and sift through rocks to find what? Gold. Today, there's whole companies that go out into the desert in Nevada and throughout the country, and they collect tons of earth, and they find a way to take all this earth and separate out the gold, the tiny bit of gold that they find in the earth. So the whole point is we need ways of separating things. We need ways of efficiently separating the glucose from the red blood cells, the red blood cells from the solution, the potassium from the solution. In this case, we're going to use something called paper chromatography. I would. Chromatography paper is basically a piece of filter paper. And you cut it in a strip, as straight as you can. I put in a tip here, and I would take some tape and tape it onto the top of the strip. And you're going to take a pencil and you're going to tape that strip to the pencil. Is that clear? Yeah. You're going to then take this marker. You're going to have 3 groups. You're going to have a bunch of different markers that you're going to know. You're going to have you have a known marker that you're going to know it's a permanent marker. What do we mean by permanent marker? Say again?
Marker that can't be erased.
It can't be erased or can't, is that what we mean by permanent marker?
I mean, it's permanent. But you can take it off though.
How do you take off a permanent marker?
Rub it real hard with water.
Okay, you rub it real hard with water. Sometimes you have there's not enough space. Rubbing alcohol. All right, so you have a permanent marker. And you're gonna have a watercolor marker or a non-permanent marker. They're gonna be the same color. They're both gonna be whatever color it is. They're both gonna be that color.
Red.
Doesn't matter. So you got this color marker, it's sitting there. You make a dot on this paper. So you have one group, and you can put a dot on it. And actually, I shouldn't even put it there. So you make a dotted line with a pencil. Anything you write with a pencil, what do we know about pencils? They can erase. What else about pencils? If they're not made of lead, they're actually called graphite. But if it's not soluble in water, it won't dissolve in water. All right, so you don't have to worry. Or it doesn't dissolve well at all by polar or non-polar substances. You're gonna take this ink. You're gonna make a dot right here in the center. Is that clear to everybody? Now you're going to cut in a triangle. The reason is I like to cut in a triangle is because I like to dip this tip in the water. I don't want you can't put the whole thing in the water. If you put the whole thing in the water, you're going to have trouble. So you might even want to take and you will be judged by your results, by the way. You might want to take and make that triangle a little bigger because you want the dot to be right here, well above the water line. This is water. In one, and then another one, you can do the same thing, but you're going to use some kind of. This is what? Is water polar or non-polar?
No.
Polar. It is polar. If you did not answer that quickly, you need to get some more work in your head. You got a lot more work to do. This was a check. This was a check. If you did not say polar quickly in your mind, then you need some more work. So then over here you're going to have what? What do you think you're going to have over here? Some kind of nonpolar solvent. Some acetone, right? Some, maybe it'll probably won't be acetone because I don't want anybody getting sick. Probably be ethanol or isopropanol or something that's going to be dissolve nonpolar things. You're going to do, so you have a permanent marker, which you think is what? What do you think the permanent marker is? Which of the two do you think a permanent marker is? Come on, guys, use your heads. What do you think a permanent marker is? Is it going to be, what'd you say again? Nonpolar is correct. Good guess. Did you guess? All right, but it was a good guess.
I guess because.
Why did you say that?
Because it takes long for water to.
Because it doesn't dissolve easily in water. That's right. And you can dissolve it with nonpolar substances like that. Like dissolves like. All right. So non-permanent marker, something that dissolves easily in water, is called what? It's a polar one. All right. So this is your polar ink, and this over here is your non-polar. This is your polar ink, and this is your non-polar ink. These two you know. So what are these two groups going to be?
Wait, what?
Good, controls.
Oh.
These are going to be your control groups. How do you know? Why do I say controls? Because you know them. You know which one's polar and which one's non-polar. You're going to use these for controls. You know what the solvent is, and you know what the solute is, or the ink. Now, let's go ahead and create our control groups. First of all, what's our question? Well, let me make the question really easy. You're going to have an unknown. You have an unknown. You're going to have one that's already made for you. It's going to be on a piece of paper, a piece of filter paper. And it's going to be an unknown. Well, what are you going to do with that? How do you figure out if this is polar or non-polar?
Stick it in there to see which one.
You'll have 3 samples. You'll have three samples of each unknown. All right, 3 samples. So that you have examples to work with. So you're going to stick it in and see if water dissolves it, and if you're going to stick it in also right, and see if the acetone will dissolve it. Do you agree?
Yeah.
Seems easy enough. Okay, well, although it seems easy enough, you're not really answering the question, because if water does dissolve it and non-polar does, the question is, how do you know then that it is non-polar, that it is acetone, that acetone does dissolve it this way, that it does it in the same way that this is doing it? How would you know that? You need your control groups, right? So you take and you are going to take your negative control. You need a negative control. And you need a positive control. Go ahead. What would you do?
Put that one, the unknown in the middle, and see which one is closest to.
And you have your experimental group. I like the way you think. I'm running out of room, so you have 3 groups, right? Your positive control, your 2 control groups, and your experimental group. What is going to be your negative control?
Permanent marker.
Permanent marker in what? Permanent marker in acetone is the positive control. Why is permanent marker in acetone a positive control? Because you know it's going to work. It's going to dissolve it. It better dissolve it. If it doesn't dissolve it, then you know there's something wrong. So that's good. See, that's the whole point of a control, is you want to compare it. If it doesn't work, then you're like, okay, there's something wrong. I don't know what it is. The acetone might not be acetone. There might be something wrong with the marker. Maybe the marker's weird, like graphite. I don't know. But there's something wrong. So this is what you're going to use so your positive control is going to be the permanent marker in acetone, or notice I'm using abbreviations here, or it'll be the non-permanent marker in water. One of the two is going to happen. All right. Either one of these is your positive control. Is everybody okay with that? Mm-hmm. All right. And obviously these are shorter than you want. The longer these are, the better they're going to work. All right. So what's going to be your negative control then? Permanent marker in water, or non-permanent in acetone. Are we clear? Mm-hmm. You know this one should not work. You know this one should. You're going to compare whatever happens with this experimental group to both of those. With your experimental groups, how many do you absolutely need?
2.
You need 2. What are those 2? Acetone, another one in water. Remember, they'll be in beakers.
I'm smart.
You are smart. You're absolutely right. So you have acetone, you have the water. Remember, you don't want the water level all the way up to the top. Nor do you want the acetone all the way up to the top. You only want it to the triangle. You want the triangle to dip in, dip the tip in, and let the water go up. Are we clear on this?
Yeah.
Now let's talk about what we're going to see and why we're going to see it. See, it's not all cakes and ice cream. Oh, I like cake and ice cream. I had the best cupcakes in my yesterday. I picked one. It's delicious. Soft. Sam's Club. Amazing cupcakes. Whipped cream frosting, sprinkles. It's the best. All right, so anyways, so what do we expect to happen and why does it happen? Who is taking PBS? All of you should be able to come up here and explain this to me. I don't need to discuss anymore. I think all I need to know at this point. Say again? I saw you. I went into your classroom. I saw you cutting out little bands, gel electrophoresis. Yeah, you know all about this. You guys are gonna come up here and explain this, right?
Yeah.
So explain it.
Yeah,
Like, what is it? How are we separate? What are we doing? What are we separating this by? How does it get separated? What's going on?
Okay, we separate the DNA and
No, this is not DNA though. You have to apply your knowledge you learned. You can't just memorize. See, this is my example. Understanding the process allows you to switch gears, go from one class to the next, and you're able to apply what you learned. So let's apply what you learned in PBS. So filter paper, much like agarose paper, or agarose, no, it's not paper, but it's your agarose gel. I'm assuming you talked about an agarose gel in your electrophoresis lab. Filter paper does this. It creates a web, a maze. Did you talk about that? How agarose makes a maze? No. Agarose does the same thing, except it uses a polymer, a protein, but it makes a maze of. Yes, please. It makes a maze, a bunch of little tunnels. And when you have a molecule, in the case that you were looking at DNA. Tomorrow you're going to be looking at ink, something called dye, right? Little molecules of colored molecules. I'm gonna shape it. You know what? I'm gonna shape it a little weird because these are all weirdly shaped. Each molecule is gonna have its own different shape. Now, because this is paper chromatography, we're not going to focus on this. If we're doing an agarose gel, the agarose gel separates it by size more than anything else. But paper chromatography is a little more complicated. But let's look at size and let's think about how the process works anyways. It certainly works this way with agarose. Those of you that don't know what agarose gel is yet, it's basically seaweed protein. And have you ever heard of jello gelatin? Have you ever eaten jello gelatin? If you're Muslim, you probably have never eaten it. If you're Jewish, you've probably never eaten it. The thing is, it's made of pig, gelatin's made of pig bone marrow. It's a protein that it's tasteless. It doesn't taste like, obviously it doesn't taste like bacon. But the gel, once you get that protein in water, it makes a matrix. Agarose is a seaweed protein, so you could actually eat if you don't like the pig bone marrow, you could use agarose. Agarose does the same thing, makes a gel. Actually, it makes a tighter gel. The more agarose you use in the mix, the tighter the gel. It's not as I don't know, what word do you call it? The wobbly as gelatin is. So, but the key is that there's little tunnels through this. And you put these in, so this ink is in a solution. And if you put a dot, by the way, do you understand the idea that when you have a color of ink, it's not one color, it's not one molecule. If you have red, there's probably a little bit of blue in there, a little bit of blue, and a little bit of green. I mean, there's all kinds of colors go into making the color that you see in the pigment. Many molecules come together to make that one color. Now, I used to go into describing how color works, the additive and subtractive, his eyes are rolling back in his head. It's so weird. You look like you're possessed, dude. Don't do that. It freaks me out. If you're going to go to sleep, go to the office and lay down. Ugh. So you put this ink, the water, or this is on paper, right? So this is your ink dot right here. Your ink dot's made of 3 pigments. One is what? Describe this dot, this pigment.
Small.
It's small. One is
Medium.
and one is right? This all makes that color. If it's black, it's made of these 3 or 4 or 5 different molecules. Are we clear? When you cut up DNA, you're making a bunch of different fragments. Same process. You put it in some kind of solution. Now, when you do electrophoresis, you're using electricity to move it up the agarose. But here, we're not using electricity. And by the way, all of us will be doing an agarose electrophoresis gel in this class. We're all creating our own gels, and we're going to be running it. We have the machinery, we have the equipment. We're going to do it. All right. So you should be paying extra attention to paper chromatography. So you stick this. This is paper. In the case of our lab tomorrow, this is paper. So if you think about this as being the paper, this is what paper. If you look at it carefully with a magnifying glass, this is what paper looks like. This is the dot on the paper. Do you see it? Does that make sense? It's not well done, but this is the dot on the paper that you put. Here's the dotted line and pencil mark, right? This is all paper. Now, as the solvent is here. That's where your line is. That's where the water is, or the acetone is. What's going to happen is these water molecules, remember, are going to move up the paper. How are they moving up the paper? I hate when people. That's the one thing. It's, you have pet peeves, things that really bother you. Like some people are bothered when you hear ticking. Other people are bothered when they hear goop. Right? You can see their faces. There's some people you can see who's bothered by it. Other people are bothered when you hear snot. Right? It bothers you. Yeah? You know what bothers me? When I ask a question, what makes this thing go up? And somebody always says, one person, every group always says, gravity. Gravity always pulls down. You never fall up. It never happens. So, okay, so it's not gravity. It's not air, it's hydrogen bonding. Thank you, somebody was paying attention. So this is going to be hydrogen bonding.
People say that.
So here's this ink with these 3 molecules. This is going to keep moving up. Somebody the door's closed, I'm assuming. This is going to keep moving up. As soon as it hits this, it's either going to do one of 2 things.
Stay.
It's going to what? Well, no, the solvent's going to keep moving out because that's hydrogen bonding. This is solvent, hydrogen bonding with the fibers of the paper. That's happening. The water's moving up, or the acetone's moving up. That's happening. The question is
it going to stay?
Is what's going to stay?
Is the ink going to stay?
Is the ink going to stay? What would make it move? If the solvent does what? If it dissolves it or not? So the first question you have to ask is you're going to find out, which is going to answer your question, right? The whole experiment is: will this solvent dissolve this ink? Yes or no, right? Either if it dissolves it, what's it gonna do?
Disappear.
No.
Oh no, it's curious.
If the solvent dissolves the ink, what's the water and the solvent's still going up? What's the ink gonna do?
Go up to
It's gonna go up with it. The ink will go up with it. It's in solution. That's the point. Will they all three stay together?
no.
Why not? I see some people giggling and talking. I'm gonna post this as it is, so when some folks ask me, why doesn't my kid get it? It's gonna be like, well, they're just not paying attention in class. This is not difficult. So it's in solution, like you would salt water, whatever. These three are going to move up with the water, or acetone, whatever it is. What's going to happen as it tries to go through this maze of fibers? Why? Look at it. I'm not asking because I expect you to be geniuses. Although I know you're really smart. That's not why I'm asking. I'm asking because it's obvious. If you look at it and think about the process, understand what it is we're talking about, and the solution, that what's going to happen is obvious. So you have these three inks, these three molecules. They're in solution, so they're going to move with the water. The water the solvent's moving up. These three, the water's small. It's polar, it's moving up through these holes, in between the fibers, very easily. These three are going to move up at three different rates. Why? This is not actually 100% accurate, but we're going to go ahead and use this as an explanation. Yeah?
Because one is smaller or bigger than the other.
Thank you. One is smaller or bigger than the other. Because if you have this giant maze of things to get through, all these fibers, you got to get through the fiber. And you're small, are you going to go faster or slower than the biggest one?
Faster.
And the bigger ones are going to go not as fast. So over time, I hope you're paying attention, although I have my doubts. Over time, what you'd expect to see is a piece of paper that's wet. And how are you going to know it's wet? You know what wet paper looks like, I hope. Where you started with a black dot here, that's where you started. But over time, what's going to happen is you're going to get a smear. It's going to be a little red, a little blue, a little green. I don't know what colors, whatever. Because to be honest, to make black, it takes a lot to make black. And this will smear out. And you'll see a difference. So you'd expect which one of these to be the biggest. Probably the red's the biggest, and probably the green's the smallest. Now, this story is actually much more complicated, but this example works really well for us when we start to do electrophoresis. Yeah, question? Yeah. All right. So that's paper chromatography. That's the process. That's the background. But now remember, what do we want to do with qualitative data? So far we've been talking about qualitative data. This one's bigger, this one's smaller, right? At best you have rank data. Can't we come up with a way to quantify this? Look at your paper. The answer is yes. We know that this ink is not going to move faster than the solvent. That's the acetone or the water, whatever it is. That solvent line is here. Everything else is going to move with it. Are we good with that idea? You have to be good with that this is the solvent line. So now let's take a look at the data here. The other says, Yeah, this dot's gonna move, the dot's gonna move, it's gonna smear. This, there's a distance. Let's say we stop it. By the way, we have to stop it before it gets to the top. If you wait to the top, you have to be able to measure how far the solvent moved. Is that clear? So if you wait till after it reaches the pencil, you can't tell how, the water's just gonna keep moving up. You won't be able to tell how far the water moved up. So you have to stop it before it gets up there. In fact, I'd stop it till when it gets about here. The longer this is, the better separation you're gonna get, for the record. You don't have to use a pencil. You can come up with your own way if you feel like you can do better. So in any case, where's the start?
We'll just start at what?
Where's it? You're gonna measure a start and an end. The solvent line where you pulled it out, that's what?
The end.
That's the end or the stop,
So the top
right?
of the triangle is the start.
Right. This, where that pencil line you drew, remember I said you draw a pencil line? That's your what? That's your start. That's your start. So, the distance traveled by who has Mr. Custer for class 10? All right. So we know how to calculate distance, right? Displacement. No. Final minus initial. You didn't learn that? Well, I'm sure you will. Final minus initial. That is how you calculate the difference between anything. So if your final is measuring from here to here, your final is 10 centimeters, your initial 0 centimeters, what's your distance traveled? 10 centimeters. Are we clear? I can't make it any simpler than that. You're just using that math. Obviously, you can use a calculator. So you move that. That's the distance traveled by the spot, and the distance traveled by the solvent is obviously good. So this is the stop for the spot. This is a solvent stop. Do you see that? Do you see that there's two stops, one for the dot and one for the solvent? We don't expect the dot to move as quickly as the solvent. The water or the acetone are small, we expect them to move quickly. The dot, we expect to move slower. Are we clear?
Yeah.
You're putting stuff away, you're getting up, you're walking out, then you're going to say, I didn't understand this, I didn't understand that. You have some questions there? You have some questions there you need to answer?
Just that one page?
Yeah, there's a one page. You might not be able to answer all of them, but you should be able to answer a lot of them. I would be answering those questions. Those are your pre-lab questions. You're going to collect your data tomorrow, and then you're doing your lab report. I expect this is part of your background. All this was your background. You can take notes on it, write it up any way you want. You have a question, right? You have a question. You're going to come up with a hypothesis. You have your control groups, your experimental groups. You're going to come up with a conclusion. You're going to make sure you have evidence. You're going to make a claim and collect evidence, state your evidence and your reasoning. You're on your own for this one. All right, that's tomorrow. So when we're talking about RF value, RF, you're going to take how far the solvent stopped, right? RF is equal to how far the dot traveled, the spot traveled. And you're going to divide it by what? The solvent travel. So the spot travels a certain distance from here, maybe it stops right there, divided by the total. That's going to give you a number. It could be anything. Maybe the RF value for the green. The RF value is I don't know, 0.5, and the red, the RF value is 0.2. Which one was a bigger molecule? The red was bigger, right? We don't have actual molecular weights. We will when we do DNA and RNA and proteins, but because you have a standard, you have a control group that you can use to compare it to. But in this case, we can do relative sizes. The R value will let us know which one's bigger, which one's smaller, which one moved further. Okay, on this paper, it gives us a way to quantify, right? To make something that at best would be ranked first or second, right? This was first, this is second, this is third. Instead of doing that, this R value gives us a number we can actually compare. Does that make sense? We're quantifying data that could be qualitative otherwise. Are we all good with everything here so far? So here's the deal I'll make you. I'll move the deadline for all the Khan Academy homework to Friday.
I thought it was already.
No.
Thursday.
Yeah.
So all of us do Friday by beginning of class. If you have problems getting on it, you need to see me. After school today, we'll work through it. All right. Wrap it up then. So what you need to do then, once you're done collecting the RF values, you need to make a table because you're going to have to graph it, right? Maybe you graph it, maybe you don't, I don't know. So you're going to need a table. What I would do is I would make a table that has several column headings. You're going to want to have what is it? What's the sample name? Is it unknown 1, unknown 2? Is it the known, polar, non-polar? I would have a column with, so here's, let's say, unknown 1. And let's say you say dots. And I would say if it separates, then you get, red, yellow, blue, right? And then RF values for each one, right? So the red is 0.5, this is 0.3, this is 0.2. So now you have some stuff you can graph. Now you could do that with all of them. You can do that with everything that you have data with. Maybe sample UK1 in water, unknown 1 in acetone, right? Nothing. Does that make sense? So that's the kind of table you'd want to make. Something like that. That's going to give you data, information. Maybe you have a column. Maybe you have a column for observations, errors. Maybe you have an error column. I don't know. It's up to you. Comments. Maybe you call this comments.
What does that say?
So you have an error column. You have comments. So here you write, hey, I saw that the ink did this and the ink did that, or it didn't split up, it moved, but it stayed as one dot. I don't know, whatever it is you want to describe and discuss. May you identify an error. Are we good? Do we understand this? Because you're not going to have a chance to discuss it with me tomorrow. Does anybody have any questions?
So, if you're going.
Yeah? Question?
Yes.
Yeah.
So you want us to take notes on the lab, but I thought we had to have everything up.
I'm sorry, say that again, you trailed off.
You want us to take notes on the lab, but I thought we weren't allowed to have like our notebooks and stuff.
You could have paper, just bring paper with you. You don't have to bring your whole notebook. On lab day, bring your goggles, a pencil, and gloves, and some paper so you can write on. The less you bring in, the better it is for you, so you're not spilling anything on it. You can bring so I used to do a composition notebook, but honestly, if you drip something on your composition notebook, it can mess it up too. You can throw a sheet of paper away easily enough. Hey, just to be clear, this is your pre-lab. This is your notes. We did most of the lab write-up here for you already, so I should have no excuses. Just to be clear, the negative control should be permanent marker in acetone, non-permanent marker in water should be the positive control, right? Permanent marker in acetone or non-permanent marker in water should be acetone. So that should be the positive control. And the negative control should be permanent marker in water and non-permanent marker in acetone. As long as we're clear that that's what it should be for your positive control and your negative control. All right.